Thank you so much for coming. It's no surprise, I think, given the discussions about digital sequence information that we wanted to put on an event here to look into how the gene banks from the CGIAR centers, so there are 11 gene banks, 11 centers have international collections, which they manage under the auspices of the International Treaty on Plant Genetic Resources for Food and Agriculture. And we are making, of course, increasing use of digital sequence information and the way in which we manage those collections. So the point of this side event is to share some information with you about how we're doing that as part of an ongoing discussion, contribution we hope to be making to a discussion about how DSI is used, how it's useful, how it generates benefits, and ultimately how CGIAR centers can contribute to efforts to share capacity as part of an evolving global system for conservation and use of plant genetic resources. Excuse me, So today we have two speakers in personam. Our first speaker today will be Marie-Noëlle Njodjop, who's the gene bank manager from Africa Rice. And she'll be talking about providing an overall introduction to how DSI is used for gene bank management. Then we'll have two case studies. The first will be provided by Carolina Sansaloni, who's the gene bank manager at CIMMIT. She'll be piping in live from Mexico, and she'll be looking at how they use digital sequence information to analyze the structure of their international wheat collection, identifying redundancies and gaps. Then we'll have a second case study from Monica Caraval, who's running the digital sequence lab that's associated with and part of the CIAT maintained gene bank in Colombia. Thereafter, we'll have a pre-recorded presentation by Mathieu Rouard on managing genotypic data across the CGIAR. And then we'll have a final presentation by Fiona Hay on a new guidance note that's been adopted by the upper echelon of the CGIAR for improving the accession level management of our collections and tying back to how we're using digital sequence information for that improved accession level management and for making strategic decisions about what materials will be curated in what different ways within the gene banks. So without further ado then, I turn to Marie-Noëlle and she will make the first presentation.
Thank you, Michael. Good afternoon, ladies and gentlemen. I'm happy to be here today to share some information that we have on the use of the digital sequence information for analyzing the gene bank collection and making conservation-related decision. The digital sequence information refers to the genetic information that is derived from the nucleic acid sequence, such as DNA, RNA, and stored and analyzed using the digital technology. It can be used in a different way. And I just listed here some few examples that can be used to identify species, subspecies. We listened this morning in the presentation that there are lack of taxonomists in the world. We have less expert in that domain. So digital sequence information can play a role here, understanding genetic diversity, identifying duplicate, guiding crop collection management, and also breeding. With the integration of the digital sequence information in the gene bank management, we have opened a new opportunity for conservation, for research, and for sustainable use of the plant genetic resources, because it has enabled the gene bank manager to work more efficiently to identify genetic resources with greater precision and develop also by breeder to develop new crop varieties that are more resilient to changing environmental conditions. So there are several techniques for using DSI in gene bank management. You have the whole genome sequence, you have the targeting sequence, you have also different platforms that have been developed in the world that use those information also. And you have several future use of the digital sequence information on the gene bank management in terms of predicting breeding, improving the efficiency and the sustainability of the breeding program, identifying novel genes on allele, or conservation of breeding program. So some application of the digital sequence information for analyzing the collection could be like species identification, taxonomy classification, phylogenetics analysis, population genetics, genome analysis. You can use genetic diversity, the analysis of genetic diversity to identify underrepresented species in the collection and prioritize the collecting effort for population that have less diverse diversity. You can use it also to identify novel genes, for trait discovery in the seed bank, like I was saying, to monitor the changes in the genetic diversity of the population over times, and also on the phylogenetics analysis. So I just want to give some few examples where we were able to use this digital sequence information to identify some human error or some misclassified species. This error could happen in the gene bank collection. If you look at this graph, where we have several groups, we found that one wild species named Oriza Bataille were able to plow with some cultivated rice. So we were able to identify those misclassified species using these informations. So from there, we were able to use to identify or to develop diagnostic marker that could be able to differentiate the species of the in the African rice collections. So we have several type of markers we developed, and those markers are able to separate the African species compared to the Asian species, etc. So we can also use this DSI to identify all the species that you have in the collection. This is the case of rice, where you have Glaberrima, African rice, Asian rice, and the wild species like Bataille and Longistaminata. You have several clusters, and you can also be able to classify them. We use them also for the sampling methodology for selfing crop like rice, to know if for the large scale characterization, if we can use 1 seed or 15 seed of a bulk of 5 to 10 seed. And based on the result, we were able to make a decision on how we should handle this large scale characterization of the germ plasma. We also use digital sequence information to determine the population structure and understand the diversity in the collection. So we have here the structure that we were able to determine, and in the specific subgroup also, we were able to see how the specific subgroup is structured. In these are African sativa compared to Asian rice, the rice domesticated in Asia, and to see how the African sativa are structured and compared to the Asian rice. So we also use this information to develop mini-core collection. You know that the goal of the gene bank is to conserve the germ plasma for sustainable use, mostly in pre-breeding and breeding, to develop new crop which are adapted to the environmental condition. So it's often very complicated to breeder to screen those large size of collection in their breeding program. Therefore, it's very useful to create what we call mini-core collections. The size of the collection can be handled easily in the multi-location testing or for spheral phenotyping by the expert. So we developed several mini-core collections for African species and also for African sativa, and they are very useful. They are now screened worldwide to identify new trait, donor for new trait. And so in the breeding program, those mini-core have been used for this trait. I just listed few traits which are important. They occur in the farmer field to identify new donor, to identify gene, and to be able to develop improved varieties. So I just wanted also to share with you some centralized database where this information are stored. I took the example of National Center for Biotechnology Information, NCBI. They have all this information where scientists can go and use the information and apply them in their program. We also have Grameen database developed by Dr. Suzanne Makouche from Cornell University, very useful for the gray species and for scientists working on the genomic research also. So this information, digital sequence information, are also very important to curate the passport data. Like I told you, when you have some problem of taxonomy, you don't exactly know the species, what you have collected in the field. If you have marker information, then those marker information will help you to classify the genetic resources that you have collected and to minimize error in your passport data. So I just want to share also some publication that we made based on this information. And with that, I will thank you for your attention. Thank you.
Carolina, can you hear us and can you take control of the slides?
Hello.
We can hear you. That's fine.
No, I cannot control the slide. Sorry. You can control for me if it's okay.
Slide control, which link?
Carolina.
Carolina.
Yeah.
This is the just, okay.
Yeah. Maybe can you boost your volume?
Oh, yes, yes. Okay.
Okay,
Yeah.
Carolina, we're ready.
Okay. Okay, good afternoon, everyone, and thanks for inviting me to give this brief talk. I will try to be very brief to show you some of the uses that we apply DSI in gene bank collection, as Maria Noel mentioned before. Well, we all know that a large proportion of the valuable genetic variation is still hidden in gene bank collection. Based on a study we performed a few years back, we found that around 8% of the collection has been used by breeding program. Means that a large proportion is still hidden, as I mentioned, while the challenge for crop production become larger. And for example, in CIMMYT, we have two of the largest maize and wheat gene bank collection, having 29,000 accession of maize and 140,000 accession of wheat. But before using, we need to understand the collection, no? And the ideally will be phenotype the whole collection. But of course, the costs are big, it's a lot of time consuming, and we have interaction with the environment. And by genotyping, by creating digital sequence information, we reduce the cost, we reduce the time as well, and we don't have any interaction with the environment. So means that DSI allow comparing material at molecular level more efficiently. This is an example of a study we performed in the wheat collection. What you can see here is a diversity analysis of all only domesticated bred wheat. We include 60,000 accessions. So each of the dots you can see here represent one accession of the collection. You can also see the structure and the composition of the collection. So now we can understand better what we concerned for so many years. So what proportion of elite background we have, what proportion of land races, and from where these land races come from. So diversity analysis stress a role of key founding varieties in modern breeding program, and also identify land races with potential value in breeding. Another important activities we did with using DSI is the curation of the passport information. As Maria-Noel mentioned before, also another example. This is in wheat. In red, for example, you can see the hexaploid wheat, in green tetraploid wheat, and in blue, the wild relative. But you can also observe some red dots across the green. Means that there is an error in the passport data, that many accessions were tetraploid, but genetically were hexaploid. So we managed to fix all this passport information based on markers on the genome. So wheat has A, B, and D genome. If we have an accession with only A and B, of course, it's not an hexaploid, it's a tetraploid accession. So all this information allow us to fix all this problem in the collection. Other important use, for example, DSI allows identification of redundancy within and across collection, no? Within, for example, only in the CIMID with collection, but also between collection. We also genotype, for example, the CARDA collection, and we can compare across collection, as also Monica will explain later, different institutions conserving same crop can compare and identify potential redundancy or some unique material as well. And conservation gap analysis to predict the distribution and quantify also the current ex situ conservation status. One very recent activities we are doing as well is mining useful alleles for climate change adaptation for the CIMID with gene bank. So having all this diversity stored, conserved in the gene banks, and having also genotyping information. Currently, we have approximately 70% of the collection already genetically characterized. Using all this information and curating the passport information, so we know exactly from where these accessions were collected. So we know, sorry, it's moving. So we know exactly the geolocation. Having this geolocation, we can extract climate information for free. So a combination of the climate information together with genotypic data, we can identify genomic region controlling different traits, important traits for climate adaptation. Some of the traits that we can work is drought, temperature stress, acid soil, salinity, fertility, agronomic practice adaptations. These are some of them traits that we can work. So it's very relevant for today's and future challenge. So we could say that before DSI, it was really, really hard to go into the gene bank and find something useful efficiently. We've been using for a long time the gene bank, but yeah, it takes time to characterize, to evaluate and select the appropriate accession. Now, with all the information we have, we can manage differently. So I believe it's a new era for the gene bank using DSI, and we can search more efficiently and find the right accession for different purposes, for climate change, for food security, for different purpose. We can target more efficiently the accession we need or relevant for the problem we are facing. And also we are using DSI in capacity building. So we organize a Latin American community of practice organized by CIMIT and CIAT. Monica is here as well. She's one of the organizers as well. And we use genomic and digital tools to enhance the conservation of genetic resources. So we created this community of practice for Latin America, and we introduced them the use of DSI to manage the collection. So in this community participate 14 countries and more than 33 institutions. So it's not only for CGIAR gene bank management, but also we like to distribute the knowledge in our case in Latin America for all national gene banks. And with this, thank you.
Thank you very much, Carolina, and thanks very much for starting this presentation at 5:30 a.m. your time. An extra hand for Carolina for getting up so early.
It's a good way to start the day.
And
thanks.
And it's 6:30 in the morning for Monaco. So also in advance for Monica.
Thank you. Good morning, everyone. My name is Monica Carvajal. I'm a scientist working at the Genetic Resource Program of the Alliance Biodiversity International and CIAT, which is based in Cali, Colombia. And I'm going to give present a case study on the use of DSI to compare international cassava collections, which are hosted by CIAT and by IETA. So this is a work that has been jointly performed with IETA. So I think Carolina and Marie-Noël has already explained that DSI help us shed some light on the genetic relationships among the accessions that are conserved in the gene banks. And this is important because it facilitates germplasm curation and also enhance the use of diversity for crop improvement. So cassava is an important tropical root crop because its starchy roots are a major source of dietary energy for more than 500 million people in the tropics. So most of the crop diversity is conserved in 2 international gene banks. So one comprises almost 6,000 accessions and is conserved at the Alliance Biodiversity and CIAT. And the other collection is conserved by IETA in Africa, and it's comprised mostly by a bit over 4,000 accessions. So the Alliance collection represents the diversity of the center of domestication in Latin America. And as you can see in the pie chart, it's mostly composed by land races, but we also conserve some improved varieties, which are product of the breeding programs and cassava wild relatives. So since 2016 and from 2016 to 2021, so we have been collecting tissue and extracted DNA from each of the plantlets accessions that we conserve. So we conserve cassava in vitro. And we have used DART-seq as genotyping platform to produce the DSI for the cassava collection. So we have managed to complete 97% of the collection being genotyped. So with DSI, as it has been already shown, allow us to perform different kind of studies. We wanted to first have a view of what is the genetic diversity, how is the diversity of the collection that we conserve. So Carolina already explained, so this is a diagram that represents the genetic diversity among accessions. So each dot represents an accession, And as you can, the closer the dots are, the more similar they are, and the separated they are, the more distant. So you can clearly see a big cloud in the middle that represents the cultivated land races and improved varieties of cassava. And there are some more distant groups represented in green color, which are the wild relatives that are, of course, farther apart from the cultivated cassava, but we see some green dots that are closer, which represent those subspecies of cassava that contributed or are closely related to the cultivated version. So taking advantage that IATA has also genotype or produced DSI information from their collection using the same genotyping platform. So we thought it would be very interesting to study the two collections together. So these pie charts show how is the composition of the two of IATA collection. So they conserve in two sites cassava, but you can see that the majority represents breeding materials and some land races. So we, in 2022, so we co-analyze or put the two DSI of the two collections together, and we perform a joint diversity analysis. As you can see in the table, you can see how was the composition of the samples that were included in this analysis. So it was very interesting. As you can see in the diagram, orange dots represent the accessions conserved by IATA, and green dots represents the accession conserved by us in CGIAR. So you can see that there is a clear complementarity of the clear genetic complementarity between the two collections. So this is in addition to being very nice, that the two collections are complementarity, and they have complementarity. There is a bit of genetic redundancy across the two collections. So this is a good example of how DSI can also aid conservation management, can support conservation management and facilitate the access and use of crop diversity. So with this study, we are producing a knowledge base for a coordinated stratified curation of the two collections. So we have been working on identifying the genetic duplicates or the genetic redundance within and across collections. This is important because we can optimize the conservation costs. Conserving plants in vitro is costly. It also is important to identify those possible redundant accessions, because if there is any germplasm evaluation or characterization, so we can optimize the resources invest for that. It's also helping us to guide the efforts that we want to invest for cryopreservation of the cassava collection, as mentioned as well by the other colleagues. So we can also use this information to identify gaps in the collection. And we are in the process of redefining new core collections within and across the two gene banks. So Carolina also gave an example of how we can make more use and support crop improvement with the DSI that we are generating. And we are also participating in this multi-center project, which is led by CIMMYT and funded by Bill and Melinda Gates Foundation. So we are working together with IATA, leveraging the existing DSI and mixing this information with the bioclimatic variables that are extracted using the geolocation data of collection sites of the accessions. And we are performing, as Carolina explained, the environmental association analysis. So we have identified some possible candidates, allele that are candidates that are under revision. And so I think it's important to also have perspective on how this work can help us in the collaboration with other gene banks. So we think that this is the way to start exploring the collection. So we have been in contact also with ILRI, which also conserve tropical forages as we do. We have some common species that we conserve. We both agreed to use the same genome-wide genotyping platform. And we are going to work in a similar way as we did with IATA to understand the Brachiaria, also known as Urochloa, and the Panicum, known as well as Megapyrsus, the collections that the two institutions concern. So with that, I finish, and thank you. So this is some of the people that contributed with this work and funding institutions. Thank you.
So against all odds, we're actually a little bit ahead of time. So we could pause for a moment if you have questions or comments that you'd like to make. Yes, John.
Thank you very much. Well, I'm also capable of shouting. Thank you very much for those beautiful presentations. My question probably goes to Maria more than anybody else because it's focusing on core collections, establishing core collections. Considering that a lot of germplasm that we collect from the farming systems, from the natural ecosystems is very heterogeneous, okay? I'm told that when this process is being undertaken, of analyzing the material using the DSI, you use individual plants. Now, my question is, how do you guard against losing information simply because you are using one individual from a heterogeneous collection?
Thank you, John, for the question. Yes, it's true that the material that we collect from the farmer field are diverse. This is why we really, the difference between what the gene bank is conserving compared to what the breeder are developing, they develop pure line, why the gene bank goes for diversity. And of course, when we collect the material in the farmer field, they are diverse, and this is what we also like, because each of the single seed can also have a specific trait. But in the case of rice, when we have a specific way to handle acquisition in the gene bank. So we look at the material, of course, and then we walk through them to ensure that in our base collection we keep the diversity if there are existing diversity in the material that we have collected. So we have the way to keep that material. Then for the genotyping, we have to, of course, do the selfing to purify, because you have to do some homework to clean not cleaning, we are not throwing away something, but just selfing to ensure that the material that we analyze now we look at the material to see if there are diverse or if there are homogeneous at the beginning. We do that work. If there are homogeneous, then we will go for one plant, like I show. If there are heterogeneous, we can bulk. We will bulk them. But you have seen on the presentation that even when we use one seed or we use many seed, we will get most of the time we get the same result. I hope I address it. So in short, we take into account the diversity of what we found in the farmer field. So we look at it before we start doing the genotyping to see if the material, the accession on which we will be working is heterogeneous or homogeneous. If it's homogeneous, then one plant can respond. If it's heterogeneous, we will do the bulk that will represent that accession in our work.
Yeah, thank you. I'm Carlo Fadda from the Alliance of Biodiversity International in SIAT. I have maybe Yeah, two questions. One relates to, I didn't understand from conservation purposes, which is the minimum number of seeds per accession that would make it kind of, viable in terms of conservation. And then maybe one that somehow relates to the national gene bank and what John was mentioning, and that is maybe thinking on how the because when I work with national system, and I've been working with Kenya, Uganda, Ethiopia, mainly in Africa, on a number of crops, and one of the issues is that very little portion of the collection has been characterized. And so when you go into, for example, identifying homogeneity of accessions, it's a big work because you don't know how much they are heterogeneous. You have to put them in the field, and then you have to wait until you can separate if there are different genotypes or phenotypes in the collection. And so how would you see the contribution of the CGIAR, so I'm asking also myself, to support national systems in terms of making sure that their collections are treated in a way with what the CGIAR is doing, and which is great. And secondly, how can we make sure that the data that they produce actually contribute to these large data sets? I was actually quite impressed to see how complementary the collection of the Alliance and ITWG-PGRFA. I thought that there would have been much more homogeneity in those collections.
Before we answer, I want to know, Monica and Carolina, can you hear the questions? And please wade in if you'd like.
Yes, we can. Thank you.
Yeah.
Okay.
The first question was conservation. What is the quantity of seed that we hold? Per accessions. Yes, we work collectively to develop some standard operating procedure in the gene bank level. So we hold in our active collection at least 800 gram of seed in the active collection, and in the for the long-term storage, we have a minimum of 100 gram of seed per accession that we conserve for rice, at least. Yeah.
Here's a clarification question. I think so, yes.
I'm Theo van Huntem, working at CGN in the Netherlands. Depends very much on the crop, of course. If you say grams and think of lettuce, it's something else than Phytia faba, right? The crux is in the number of plants that you use for your regeneration, because that's the genes that you contribute to the next generation. And we had the excellent presentation about cassava, which is in vitro. So you can assume, sorry, that is just one genotype. If we're talking modern varieties of lettuce, it's one genotype, so it doesn't really matter as long as you have sufficient seed to convey to the next generation. If you're talking land races, if you're talking cross-pollinating crops, the number of seeds, the number of plants contributing to the next generation must be much higher. But then again, it depends on take cabbage, where you have to hibernate and go over the winter and pollinate in isolated cages, right? Having large plants, then 50, 60 plants is as much as you can handle. And then it becomes an economical question. How can I spend my euro or my dollar in such a way that I can conserve as much alleles as possible? Your question is very relevant if it comes to the SNPs, to the genotyping, because usually we only use one plant per accession. Knowing that there's genetic diversity in the accession has huge implications. In rice, it's simple. In cassava, it's completely simple. But most other crops are taking into account the diversity in the farmer fields, creates a complexity that was not addressed in the presentation so far. So I think it was a very good question.
Yeah, and I know that Carolina's done a lot of work on this on maize, which of course is one of the more complicated crops. Having said that, we're no longer in the enviable position of having time. We've got to catch up. So I'm going to show the next video, and then we'll have the last presentation. We also have Bonnie Furman here, who we hope will be able to stay till the very end. If she can't, she can't. Yeah. Okay. Yeah. Bonnie was very much involved in the development of the guidance note that will be presented by Fiona, and would say some things about it from the perspective of the Secretariat. We'll say them for her since she's not here. Okay, we'll go ahead with the video.
Hello everyone, my name is Mathieu Roy. I work for the Alliance of Biodiversity International and CIAT, where I lead a team of bioinformaticians that are linked to the International Gene Bank on Banana. So even if I cannot be with you today, and sorry for that, I'm still very happy to have a chance to talk about the management of genotyping data in gene banks. I'm sure my colleagues Marie-Noël, Carolina, and Monica already highlighted what we mean by digital sequence information in gene banks and how important they become over the past years for conservation and use of genetic resources. But it's also very important to remind and stress that handling DSI can be also very demanding. Each time you take a plant to conduct an experiment for sequencing, whatever the machine you use, what you get in return are usually terabytes of data that are challenging to manage unless you have access to appropriate storage, analytical tools, and computing resources, as well as access to experienced bioinformaticians to help you to deal with the data. But even if you can get rid of these constraints and difficulties, what you will get at the end are genotyping data files that are very big files that are very difficult to manipulate if you don't have the proper skills in bioinformatics to do that. So what you see on the screen here is a type of file, genotyping data file, that is called a VCF, for variant call format, where you have information about your genetic markers for all your samples. And you can have in this file millions of lines. So obviously that is very difficult to have a global overview of your dataset, to explore it, and also to change it if you need. Basically what we need is, if we work in gene banks, is to collect this genotyping data, to send it to a system that would allow to store and share the data for the user of the gene bank. That can be the gene bank itself, but also multiple labs in the world, breeders, farmers, and so on. And to be able to check the data, to adapt the data to our own needs, and to export them to do subsequent analysis. To check, for instance, the diversity of your sampling, to do population genetics, to look at the genes that are close to your markers, to look at QTLs, or to look at diversity of chromosomes. So you have many examples of analysis that you can do with that. But those data in itself are not enough. What you need is also to be able to connect with metadata, like the passport data that you can find in Genesis. And this type of tool actually exists, and there is one that is called GIGUA. So we have been collaborating on this tool for many years now with CIRAD and IRD. And what it does, it stores large amount of genotyping data. So billions of markers can be stored in the database. And then it provides an online graphical interface to share the data with the users, to explore and visualize the data, to filter the data, to browse very specific markers and genotypes, and then to export those subsets in various formats and external tools. So GIGUA is open source, it's free to use, and it can be set up in any type of platforms. So let's give an example of what we mean by filtering. So I want to see what is the marker distribution along chromosomes. So I can do that. I want to know how many markers I have with a very good quality score but less than 50% of missing data, if I want to do diversity analysis, for instance. Other people would like to have a list of markers that are very discriminating between two groups of population, between wilds, between cultivated accession, or between genotypes that have been used for stress, and you want to compare with the control. So you can see exactly what are those markers in the application. Or you may want to export a very specific region on a chromosome because you know that are genes of interest, and you want to see what are the markers and visualize that in a specific tool for genetics. Or you want to know, because you know one marker of interest, you want to know what are the genes that are nearby, or if your marker is in one gene, and does it affect the structure of the gene. So everything can be actually done in GIGWA, and we have applied the system to the international gene banks for banana. And what you see on the screen is an article that we published recently where we described how we can take advantage of the system. So you can see it at the bottom of the screen. This is the type of interface that you can manipulate with GIGWA. And we compiled a number of studies that have been done for GWAS, for analysis of chromosome structure, genomic selection. So everything was put in the system, and now people can access that. It's free to access, it's open access, there is no restriction. So we are not the only one to use it. It's also being used by ICARDA, by the gene bank at ICARDA. It's being discussed for other gene banks in the CGIAR at the moment. It's also being used by national research organization in France, CIRAD and IRD, because they are very active in the development of the tool, but also in some lab at USDA. And it's also being adopted by some breeding system, because there is also the same type of needs when you want to deal with genomic selection, for instance, in breeding programs. I want to conclude this presentation by talking about capacity building, because this is really at the core of the question about DSI. How to facilitate access to genetic data if you don't have skill in bioinformatics, how to facilitate online access for many organizations, many labs. So I think GIGWA can allow that, and we have been involved in a master degree over the past years at the University of Montpellier, where we teach students how to take advantage of large genome sequence data when you want to study the diversity of important crops like rice, banana, sugarcane, and others. So for that, we took the example of the 3,000 genome of rice, and we show to students how they can export data for those 3,000 genomes that are difficult to manipulate. And this course actually raised interest in other universities around the globe, and there will be some type of adaptation courses in Senegal and Portugal this year that is led by the AGAP Institute. So with that, I would like to thank you for your attention. So if you have any question, don't hesitate to send me a message. My address is at the bottom of the slide. I'm sure also my colleagues can take a few questions, and I give example of article about the system I have described in this presentation if you want to look for more information. So thank you very much again.
Well, thank you to Mathieu, but he's wrong. We don't have time for questions right now. We have one more presentation to make, and it's a bit of a segue. We want to present now this guidance note that's been developed and adopted at the highest level in the CG for how we manage our collections and how we report on the management of our collections in terms of the curatorial status of the accessions. And there is a linkage with DSI because, of course, DSI helps inform the decisions we make about the curatorial status.
Good afternoon. My name is Fiona Hay, and I spend some of my time working for the GeneBanks initiative of the CGIAR from my base at the Aarhus University in Denmark. but I'm actually going to be presenting this today on behalf of Charlotte Leslie, who's the Senior Science Director for the GeneBanks Initiative. I will say something in particular about the guidance notes which we've developed on improving accession management, which can be informed by digital sequence information. But before we get to that, I'll just be saying something about some of our other work in the CGIAR GeneBanks. So as you know, the CGIAR have been conserving germplasm in the GeneBanks for more than 50 years. Under the GeneBanks initiative, we have 4 work packages. The first work package is about our routine operations, so conserving diversity and perpetuity. Work package 2 is about future-proofing. So this is the research that we need to do to ensure that we can continue to conserve the material in perpetuity and to improve the effectiveness and efficiency. Work package 3 is about enriching data for use, and work package 4 is about partnering with NARS. Here are some of our key results from 2022. So we have done 38 capacity development events with 625 participants from more than 50 countries. We've improved or newly drafted 49 standard operating procedures, SOPs. Georeferenced genotypic morphological data has been improved for 30 crop collections, and more than 250,000 accessions have been processed by the germplasm health units within the CGIAR. Importantly, more than 57,000 germplasm samples have been distributed in response to requests from users in different countries as well as from within the CGIAR. Here you can see where those users are based, so whether they're within the CG or universities or NARS and so on, and the countries where the requests came from. And we're still working hard to try and help our users to find the material, the accessions that will be useful to them. So for example, on digitizing plant and seed images, refining georeference data, which is very important, providing more genotypic information, subsetting tools and subsets for specific traits, phenotyping where appropriate, and in capacity building. And as you've heard, we're genotyping more and more of our collections. This is to provide new understanding of diversity within and among collections, and we've heard from Monica and Marie-Noël and Carolina, but also to identify sources of traits and allelic variation. This facilitates developing the composition and balance of collections in representing crop gene pools. It strengthens our quality control, it aids taxonomic identification, as we've seen, and it also gives the possibility of creating new kinds of materials for conservation. And this then, of course, all influences what is being requested from the CG GeneBanks. Improved understanding of diversity gives us the opportunity to reconsider and improve the composition and curation of our collections. And this is where our guidance note comes in. This is the framework which we have developed for accession curation, which is described in that guidance note. And I should say that FAO participated significantly in the development of this guidance note from inception and throughout, and specifically to ensure that international obligations would be honoured. As you can see here, I should say actually the guidance note, it is available on CG Space, and it has been submitted for publication in Plant Genetic Resources with Jean Hansen as the lead author. In this framework, you can see that we have 4 categories of accession management. The first category are those accessions that are fully curated according to our SOPs and the GeneBank standards. We then have accessions which would become partially curated. So these are accessions where we only do a subset of normal operating procedures on those accessions. So for example, we may exclude some accessions from the base collection. We then have archived material, and this is material that we would no longer monitor or distribute. And the material, the accessions that are archived, unless they're de-archived, would eventually die, and then they'd become historical accessions. So the historical accessions are accessions that no longer physically exist in the GeneBank. Decisions about accession category will be reviewed periodically. So you can see that the categorization might change among these different categories for different accessions depending on what's happening. All of this will be documented in Genesis and then through that into GLIS as well. So that's just an overview of the guidance note. And with that, I'd thank you for coming and thank you for your attention.
Yeah, we've come to the end of the time we have allotted for us and for you. I believe delegates are supposed to be back at 3 sharp. If you want to stay, maybe we can ask some questions. If you have any questions for those who want to stay, we could talk about the guidance note or any other issue. But thank you very much. Thank you so much to Monica and Carolina for joining us and getting up so early this morning.